Hello, this is Nels. Today, Jacob and I are talking about nicotine. We will be covering several topics. What is it? What can it do for us? What does it do in nature? And how is it synthesized? This is the nicotine molecule. It is comprised of a pyridine ring and a pyrrolidine ring. These are joined by a chiral center on the pyrrolidine ring. Jacob will go into more detail of the synthesis later in this presentation. This chiral center changes the toxicity and psychoactivity of the molecule. However, within tobacco plants, 99% of the nicotine molecules are formed as the S enantiomer. This molecule accounts for between 90 to 95% of the alkaloid content in tobacco. But what are alkaloids? Alkaloids are a broad group of molecules produced by plants that contain nitrogen. We can see both components for nicotine, pyrrolidine and pyridine, on the image to the right. The purposes of these compounds vary greatly. Many are defensive, attempting to kill or deter insects, bacteria, fungi, or even other plants. Humans have also found many uses for this group of molecules. Cocaine and caffeine are well-known stimulants and members of the alkaloid family. Additionally, many medical uses have been found for these molecules. They have even been used to treat heart conditions and diabetes. So now, what does nicotine do? For humans, nicotine's primary use has been as a stimulant, usually through inhalation. As it reaches the brain, it binds to acetylcholine receptors tighter than their normal ligand. Because of this, they can desensitize these receptors, increasing the tolerance for both nicotine and dopamine. Nicotine can also reinforce other stimulants, increasing the response to them. So what does the acetylcholine receptor normally do? Nicotinic acetylcholine receptors are ligand-gated ion channels, allowing ions to flow freely when a ligand or agonist binds. Most are only large enough to allow sodium and potassium to flow through, but some are configured to allow calcium ions to flow as well. They are responsible for many signaling pathways in the body. In nerves, acetylcholine can stimulate signaling pathways between cells. They can also cause a cascading release of other neurotransmitters, such as dopamine. What does it look like when nicotine takes the place of acetylcholine? Nicotine can activate several pathways that may lead to addiction and overuse. The mesolimbic pathway is a dopaminergic pathway, meaning it releases dopamine, similar to the body's normal reward system. Endogenous opioids also activate the reward system through another avenue. Nicotine also causes the release of epinephrine, or adrenaline, into the bloodstream. This is what is responsible for its stimulating effect. The normal half-life of nicotine is about 2 hours, though it can be increased by the addition of menthol. Menthol, a common additive to cigarettes, has been shown to increase the half-life of nicotine by inhibiting its metabolism, allowing its effects to be felt for longer. What happens when someone is exposed to too much nicotine, or for too long a time? Chronic use of nicotine desensitizes its receptors. Once this point is reached, the receptors will stay open for prolonged states, reducing the cell's control of its electrical potential. While small doses act as a stimulant, excessive doses begin to bind to nicotinic acetylcholine receptors in the muscle, which can lead to respiratory paralysis, the mechanism of action of its insecticide function. Speaking of that, what purpose does nicotine serve within a tobacco plant? Though we may use nicotine for pleasure, plants use it for survival. It has been shown to serve two related purposes in the plants that produce it: defense and repair. When insects consume food laced with nicotine, they go through a period of hyperactivity and then paralysis, with their tissues displaying evidence of oxygen deprivation. Nicotine also can be broken down into whatever molecules the damaged leaf needs to rebuild. This makes the molecule very useful in times of distress, but it would waste a lot of energy if it was always maintained in high concentrations. To save resources, plants have developed a way to increase nicotine synthesis when it is most needed. In the case of a single traumatic event, such as a storm, the plant moderately increases nicotine production for a short time. But when the plant is damaged several days in a row, potentially by a returning pest, it increases its production dramatically. Removal of flowers stimulates nicotine production as well as leaf growth. This is called topping. The plant also has a natural mechanism for bringing nicotine to the most valuable leaves. As nicotine travels from the roots in the xylem, it moves where the greatest gas and water exchange is occurring. Because leaves performing photosynthesis have a high gas and water exchange, more fluid containing nicotine flows into them. Thus, the plant can protect its most valuable resource. Another way the plant responds to damage is the modulation of microRNA. The removal of flowers from a tobacco plant causes the downregulation of microRNAs NTA-MIRX17 and NTA-MIRX27. These miRNA bind to mRNA and prevent it from being translated. Once these miRNA are removed, their corresponding mRNA may be translated. This allows the expression of QPT1 and 2, which are part of the synthesis of nicotine. But we still haven't answered the big question. How does a signal travel between the leaves and roots of a nicotine plant? This occurs through the jasmonic pathway, named after the primary signaling molecule, jasmonic acid. This acid is synthesized in the leaf and travels through the stem into the roots. Here it activates a protein complex called SCF-CoI1. This activated complex tags Jas proteins with ubiquitin to facilitate their degradation. These Jas proteins are now unable to bind the MYC transcription factors, which stimulate the production of enzymes that produce nicotine. Jacob will now discuss how nicotine is synthesized.
Okay, so now we can get into the biosynthesis of nicotine. And nicotine is this structure shown here below. And as we can see, there's basically two ring structures in nicotine. So the first ring structure that we have is the pyridine ring, and that's highlighted in the yellow here. Next, we also have the pyrrolidine ring, which is highlighted here. To synthesize nicotine, there's basically two pathways that are going to converge and then combine these two ring structures. So first, you synthesize the pyrrolidine ring, and then you synthesize the pyridine ring, and then those two rings are joined together. So first we're going to look at the pyrrolidine ring pathway. So when we form the pyrrolidine ring, there's basically two entry points into the pathway, and the main goal is to synthesize this molecule, putrescine. So the first way that putrescine can be made is through a direct method. So now this direct method is typically done in animals and fungi, and it starts with ornithine, which we just recently learned about. The other way to synthesize putrescine is an indirect method, which is typically done in plants, and that starts with arginine. Now, the method in tobacco plants for nicotine synthesis is going to be the arginine. So first we can look at the direct putrescine synthesis. So this starts with the molecule ornithine. Now basically what is happening here is that there's going to be a decarboxylation. This enzyme, ornithine decarboxylase, for the direct synthesis of putrescine, uses a PLP cofactor to stabilize the decarboxylation of ornithine. Basically, what happens is the amino group of ornithine, the alpha amino group, attacks the ketone area in the PLP cofactor. Now, in the actual enzyme, there's a lysine residue attached to the carbon here that is formed by an end double bond there. And then basically what happens is the amino group of ornithine attacks in and forms the shift space and decarboxylates, and you get putrescine. From here, we can look at the indirect synthesis, which is what goes on in plants. So the first molecule that we start with, as I mentioned before, is arginine. Now, the first molecule, or the first enzyme, is arginine decarboxylase. This enzyme essentially is again just decarboxylating the arginine so that we can synthesize this new molecule, agmatine. Now, this goes through the exact same mechanism as the previous enzyme with the PLP cofactor to decarboxylate. Once we have agmatine, the enzyme agmatine immunohydrolase catalyzes the formation of N-carbamyl putrescine. Now, basically what is happening in this reaction is that this amino group highlighted is becoming a ketone group. The way that this reaction goes forward is an OH group basically attacks in and kicks off the amino group after it gets deprotonated. So now we have this molecule, N-carbamyl putrescine, and the next enzyme is N-carbamyl putrescine immunohydrolase. So basically here we're trying to get rid of the amide that is highlighted over here on the left of N-carbamyl putrescine, and then that will give us putrescine. The way that this enzyme works is basically an OH group attacks the amide, the amino group leaves, and then there's a decarboxylation to give us putrescine. Now that we have putrescine, the next enzyme is putrescine N-methyltransferase. So if we remember back to nicotine, the pyrrolidine ring had this methyl group on the nitrogen. So basically this is the start of that, getting that methyl group on the nitrogen, and the putrescine N-methyltransferase uses the cofactor S-adenosyl methionine. Basically what happens is this methyl group on the SAM cofactor is able to be removed, and this nitrogen is going to do a simple add in there to add the methyl group and make N-methylputrescine. Then that adds that methyl group there. Moving on, we have N-methylputrescine oxidase, which catalyzes the removal of the amino group on the end of N-methylputrescine and turns it into an aldehyde. And then finally, we cyclize the N-methylaminobutanol that we just formed, and this happens spontaneously, and then the pyridine ring is now synthesized. So basically this nitrogen will attack in to the aldehyde, and it'll form a cyclization after a deprotonation to form the double bond here. So now we have our pyrrolidine ring that is used in the formation of nicotine. So the other part that we had to So now we have the pyrrolidine ring synthesized. Now we can look back and we can see that the other part of nicotine that had to be synthesized was the pyridine ring. So the pyridine ring, basically, the synthesis of it starts with aspartate. From here, the first reaction that's going to happen is aspartate oxidase is going to make alpha aminosuccinic acid. And this is a pretty simple reaction where we just get a deprotonation here, and it forms the imine bond to make alpha aminosuccinic acid. And in the process, an FADH2 is made. So that amine there gets turned into the imine. So now the next reaction is quinolate synthase. Now this is a pretty cool reaction, I thought, and it is the mechanism that we are going to be looking at in more depth here. So quinolate synthase basically combines alpha aminosuccinic acid and dihydroxyacetone phosphate to make quinolinic acid. Okay, so now we're going to look at this mechanism and we're going to draw it out. So the first molecule that we start with is an alpha aminosuccinic acid. And that molecule looks like this. So we have our amino group up top there. And the first step that is going to occur is that we're going to have a base. And the base is going to pluck off the hydrogen. The electrons are going to fall down and go up onto the amine group. From here, we have this molecule. And then we're going to bring in a dihydroxyacetone phosphate molecule. And what's going to happen is the lone pair is going to collapse down, and the double bond is going to attack this carbon to kick off the phosphate of the dihydroxyacetone phosphate. And so now we have these two molecules joined together. So this is basically, we're starting to get that, that ring structure that we're really trying to get after here. So this basically this portion up here is the alpha aminosuccinic acid, and then the portion I'm about to draw is the portion that's coming from the dihydroxyacetone phosphate. Okay, so then the next step is that we're basically going to be switching this ketone and this alcohol. So simple step here is we base deprotonate, form the double bond, kick up, and protonate. So now this is going to form the following molecule. So there's the OH group that we just formed. The double bond. And so to switch that group, we're going to base deprotonate here, drop down to form the aldehyde, and then protonate at this carbon. So now we have the following molecule. So now if we remember, we're trying to form this ring structure that is quinolate. So now basically, after basically rotating the molecule a little bit here, but basically what we can have happen is there is a hydrogen here right next to this carboxylic acid, and we can base deprotonate it, and we're trying to get this nitrogen here to attack the oxygen. So when we base deprotonate, the electrons go down, and then these electrons can go and attack the aldehyde, and then those go up and get protonated. So now we basically form a ring structure. Got the double bond there. And then our two alcohol groups. And then, so following this, we have this nitrogen on, this hydrogen on the nitrogen. A base can deprotonate that. Electrons fall, and then we release a water. So we're getting pretty close now. So now the final step that we're going to have is we're trying to form that conjugated ring system on the inside. And so basically we have this hydrogen right here, and the base is able to deprotonate, drop down the electrons, kick off the water. And there we now have the conjugated ring with the two carboxylic acids. That is quinolinic acid. Basically, this quinolinic acid is going to undergo a reaction with the enzyme quinolinic acid phosphoribosyltransferase, and that makes nicotinic acid mononucleotide. Then nicotinic acid mononucleotide is then able to go through the NAD synthesis pathway. So basically it's this whole pathway of forming the cofactor NAD, and in that pathway one of the products is this nicotinic acid. So now basically we have the pyrimidine ring, the pyridine ring, that we need to form nicotine. So we have the pyridine ring and the pyrrolidine ring. Next, we have to basically just join these two molecules. So now what's interesting is that the actual mechanism and the enzyme that does this joining is not known. The research basically said that nicotinic acid, somehow there was a couple theories about which intermediate is used to join to this N-methylpyrrolinium, but the actual intermediate of nicotinic acid that gets to the joining of the molecules to make nicotine is not known. So basically, once we have this pyridine and the pyrrolidine, the molecules join together and make nicotine. And that is all. Thank you guys for watching. Leave a like, subscribe. Hope you liked the video.